Okay, so this is part two of the protein, well, the translation, protein synthesis, protein processing lecture. And so we talked about the basics or the details of the actual process of translation. Remember the three main parts in terms of initiation, elongation, and termination. Now we're going to talk a little bit about regulation of translation. Remember that when we talk about regulation of gene expression, a lot of it happens on the transcriptional level, and we talked about that already, and you already tested on that. And so in terms of transcription factors binding to the DNA to basically block transcription or to increase transcription and things like that. However, there is still some regulation that happens on the level of translation as well. Okay, so it says there's translational repressor proteins and non-coding microRNAs that can basically interfere with the process. Also, if you just think about global translational activity, meaning in general, right, just sort of slowing down all translation and or increasing translation, is modulated by response to stress, right, nutrient availability, and also growth factors in the cellular or extracellular environment. Okay, so one example of regulation is regulation of ferritin. Okay, so this is a protein that stores iron. And so this is an example of regulation of translation of this specific protein by repressor proteins. Okay, so when iron is absent, there is an iron regulatory protein called IRP, which will bind to something called the iron response element in that 5' untranslated region of that mRNA, and it will block translation. Okay, so again, if iron is not available, right, there's no iron around, there's no reason for the cell to make ferritin because ferritin is a protein that stores iron. So it's only going to be made when there's iron available, okay, that needs to be stored. All right, so you can see here. Here, this is showing you the mRNA transcript, right? Here's your 5' cap, your 3' poly A tail, and this is showing you the region where that the regulatory protein here will bind. Okay, so again, if there's adequate iron, right, you can see all these initiation factors binding like we saw before, right? When we talked about initiation of translation, all of them coming in, the tRNA, right, coming in, and essentially translation is going to start, right? However, if there is no iron around or low amounts of iron which don't need to be stored, then this iron, right, regulatory protein, which we mentioned here, IRP, will bind here and basically keep this whole process from starting. Okay, so translation cannot be initiated because this essentially blocks the binding sites of these initiation factors. Okay, well, basically blocks the binding site of that 40S ribosomal subunit, but also there's other initiation factors that would come in as well. Okay, so basically that disrupts and blocks all of those proteins and the ribosomal subunit that are necessary for initiation of translation. It blocks all of them. Okay, no translation. It says here some translational repressors will bind to specific sequences in the 3' UTR. Right, so kind of showing you, this is just really saying that there are, this is just one example, right? There are other types of repressor proteins like this that might bind to other specific sequences, okay, in this 5' untranslated region of the mRNA transcript, okay? Some can actually bind to the initiation factors themselves. Okay, same idea. It's going to interfere with the interaction with the other initiation factors, and therefore translation will not occur. Okay, so that's what this is showing you here. So here's a repressor that binds right to that UTR, and again, it's going to interfere with other initiation factors being able to bind. Okay, all right. Now, these types of proteins that bind to, right, so this is showing you binding to that 3' UTR. Okay, sorry, I think I'm, so here it's binding to the 5' UTR. There are other repressors that can bind to that 3' UTR. Because again, remember we showed that there are other initiation factors that need to bind to that area as well. Okay, so that's what this is showing you. Again, it's inhibiting the whole inhibiting the ability of that whole initiation complex to form. Okay, so there are various ways in which these repressors can interfere with those associations. Okay. Okay. Another sort of function of proteins that can bind to those 3' UTRs they're also involved in basically localizing certain mRNAs to basically specific regions of cells. Okay, so this type of thing is important in eggs or embryos, right, for development, meaning that you have to sort of identify and localize specific proteins to specific areas of that developing embryo. Say, a specific area where the brain's going to develop, right? You need to make sure you can localize certain proteins there. And so apparently there are a lot of regulatory proteins involved in this process that will bind to those 3' UTRs. Okay, so this is just giving you an example, showing a specific type of protein or an mRNA for a specific type of protein here, localized right here in this vaginal cortex of, this is a frog oocyte. Okay, so again, there's many different ones. It's just giving you an example. Again, it's a binding of certain proteins to the 3' UTR in the mRNA will basically allow that protein to be localized in a specific area of a cell. Okay. All right. Now, more regulation. So all of what we just talked about in those last 2 slides were other proteins that will bind and affect translation, right? Or may also be involved. So one was kind of blocking translation, right? Another example we just gave was also being able to localize translation to a certain area, okay? So if you think about it, right, this protein, this X-clerk, would only be made in this area. It would not be made here because the mRNAs were localized there, okay? Now, another way that you can regulate gene expression, and we mentioned these things before, right, in a previous chapter, are these RNAis or RNA interference. And remember that the RNAi is mediated by other types of RNAs as well, right? The siRNAs and microRNAs. And so these actually are involved in blocking translation, essentially, okay? And so they're involved in regulation of translation, in that they can bind and block it, okay? And so there are many of these, and I think we probably said this before. And so it says each one of these, so it says as many as 1,000 microRNAs that are encoded in mammals. And each one of those can target up to 100 different mRNAs, okay? And so they play a big regulatory part, right? It says they're also important in embryonic development and may play a role in cancer and other diseases. So I think we still have a lot to learn about these, but we know that they are involved in regulation of translation, okay, in that they can bind to mRNAs and block translation or what have you, okay? So they are regulatory molecules, right, or molecules that are involved in regulating translation. Okay. Another way in which you can regulate translation. So what we just, again, if we back up, and actually if you look at let's take a look at our review sheet. So 1 second, let me just look at mine to see where we're at. Okay. Right. So remember, I'm just gonna kind of keep reminding you to draw your attention to the review sheet. So on your review sheet, obviously on page 1, we're underneath regulation of translation. We talked about repressor proteins, right? And talked about that ferritin example. And that ferritin is a protein that's regulated based on whether iron is present or not, right? And there's this particular repressor protein that will repress translation of that. We also just mentioned RNAi, right? Or siRNAs and microRNAs that are involved in regulating translation. The next part, and again, you can see this on your review sheet, is modification of initiation factors. Okay, so meaning the initiation factors themselves are going to be modified in some way, i.e. phosphorylation, and that's going to affect translation. Okay, so it's not another protein or RNA coming in to block those initiation factors from getting and binding to the mRNA. Those initiation factors themselves are going to be modified. Okay, so it says this results in global effects on overall translation activity, right? Because again, if you're going to modify a particular initiation factor, that initiation factor is involved in all translation, right? So it's not going to be specific to a certain protein, like we saw with that ferritin example, right? That ferritin example was a specific example. That repressor protein will only bind to the mRNA for ferritin. It's not going to affect any other proteins. Here, these effects on the initiation factors themselves will have a global effect on translation, okay? So again, if I asked you about, if you look at those choices under regulation of translation on your review sheet, and I asked you about which one would have a global effect on translation, the answer is modification. We're talking about that right now. Those other ones are specific, okay? They're specific to particular proteins. Here, we're talking about globally. Okay, so again, I mentioned this is phosphorylation, right? So phosphorylation of initiation factor 2, initiation factor 2B, by regulatory protein kinases. So remember, I think we mentioned this before, a protein kinase or a kinase in general is a protein that phosphorylates proteins, okay? So they are regulatory molecules. And so if it's phosphorylated, if the initiation factor is phosphorylated, it will block the exchange of the GDP for GTP. So remember I showed you that when we were talking about those initiation factors, and I showed you a diagram that showed you the initiation factor with GTP bound to it or with GDP bound to it, and that it's only active, right, when GTP is bound. Okay, so if the cell wants to regulate this, it can actually affect that process. Okay, so here it's showing you adequate growth factor supply. So there's plenty of growth. So think about your cells, right, growing in your flasks that you guys did your cell culture on. If there's adequate growth factors in the extracellular environment for you, that would mean the media, right? Then translation is going to proceed, and then the cells are happy and they're going to make all the proteins they need, and they're going to carry on, okay? So that's what this is showing you here. Translation proceeds. However, if there's stress or there's not enough growth factor in that extracellular environment, the cell is going to want to conserve energy, right? And it's going to need to slow down translation and block it in order to kind of go into sort of a dormant kind of phase to try to survive whatever that stress is, okay? And so here you can see that this is phosphorylated, okay? So if we go back here, not phosphorylated, right? And basically you can go back into its active form. However, here, if this initiation factor 2 is phosphorylated, it's no longer able to go back to its GTP form, and therefore translation is inhibited. Okay, so this is when there's some stress or there's not enough growth factor in the extracellular environment. This is what will happen. And again, the way that that's regulated is by phosphorylating an initiation factor, right? Or modifying an initiation factor. Okay. All right. So it says here's another example, right? So another example of the effect of growth factors on translation. So here's another initiation factor. Okay, let's see here. So I don't know that I'm that concerned about the details of these two, but these are two examples that have common theme. It's phosphorylation, okay? And it's phosphorylation of essentially initiation factors or something that's going to interact with those initiation factors, okay? So what this is showing you, so actually this is kind of a little bit different, but it's the idea is that phosphorylation will be able to regulate the activity of a protein and therefore the activity actually of translation. Okay, so it says growth factors will activate protein kinases that will phosphorylate regulatory proteins. Okay, so these are other proteins that bind to these initiation factors. So when the growth factors are around, these guys will bind and inhibit translation. So this is just another mechanism in terms of how translation might be inhibited when there's no growth factors around. So again, it's just a kind of a separate mechanism, but sort of along the same lines, okay? And so here in this case, right, you can see this 4E, BP, this binding protein is sitting on 4E, keeping it inactive, right? Basically not allowing any of this to happen downstream. Okay, so that means if there's no growth factor around. However, if there's enough growth factors in the extracellular environment here, what will happen is that it will lead to phosphorylation of this, which will then end up, causing some sort of conformational change and release it, right? And then once that's released, as you guys can see here, now all the other initiation factors can come in and translation will proceed, okay? So again, sort of a little bit different mechanism, right? A different variation of it, but again, it's kind of the same basic idea that you can modify certain proteins, even initiation factors themselves, by phosphorylating them, and that will then alter their activity, okay? And it can stop or inhibit translation, okay? And that phosphorylation of certain proteins is dependent on the presence of growth factors or stress in the environment. So this is one way that the cell can respond to what's happening in the environment and basically be able to slow down or inhibit translation as needed, okay? So again, it's phosphorylation or modification of those initiation factors caused by something happening in the environment, okay? That will then inhibit translation, okay? All right. Okay, so that's what we're going to say about regulation. So that ends that part. So again, if you guys look at your review sheet, we're now moving on to protein folding and processing, okay? So we talked about regulation of translation, repressor proteins that actually bind and interfere with all the other proteins getting there. We talked about siRNAs and miRNAs or RNAi that can actually kind of do the same thing, right? They'll interfere with that, with specific mRNAs to interfere with translation of that specific mRNA. And if we talk about modification of initiation factors, i.e. phosphorylation, this is going to have a more global effect. And so you should be writing that in your notes there on the review sheet. Okay, so moving on, like I said, the next part is protein folding and processing, okay? And so we have talked about, in fundamentals or general bio or bio 111, whatever you want to call it, right? We've talked about the different levels of protein structure, right? We talk about primary structure as being the polypeptide chain. Then we talked about secondary structures, right? Like beta-pleated sheets, and then tertiary structures and so on, okay? And then that 3D protein conformation or protein shape is really dependent on interactions between the side chains of the amino acids, okay? Right? And we talked about this, an experiment that showed this, that you can denature a protein, right? And if you just leave it to come, kind of let that temperature come back to, body temperature, it will refold back into its original conformation because all of that information lies within that amino acid sequence, okay? All right, so however, protein folding isn't the only thing that has to happen to proteins before they become functional proteins. There are other types of processing that can occur as well. All right, but let's first talk about the protein folding, okay? So first thing here are chaperones, okay? So chaperones are proteins themselves, but their job is to facilitate the folding of other proteins. Okay, so we've kind of just up until this point talked about proteins having to fold into a proper conformation, talking about the levels of structure, but not really talking about how that actually happens. Okay, so these chaperones, they facilitate the folding. How do they do that? They act as catalysts that assist in the self-assembly process, but they actually themselves don't become part of the folded protein. Sorry, I'm just gonna, that's my house phone, so let me. Okay. All right, so they will bind to, they'll stabilize unfolded or partially folded polypeptides. They will also bind to polypeptide chains that are still being translated on the ribosomes to keep them from folding improperly, and you'll see an example or a picture of this. Right, so that's what this is saying here, right? That chain has to be protected until the whole protein is synthesized, essentially, or until the whole polypeptide chain is synthesized, right? So if you look at this here, right? So here's your chaperone. Again, chaperones are proteins that help other proteins fold in some way. There's kind of various ways they can do it. Here's one thing that they can do. Here's your mRNA. Here's your ribosome. Here's your growing polypeptide chain. And as it's growing and amino acids are being added, it associates with these chaperones, okay? And the chaperones keep it from folding on itself before the entire polypeptide chain is made, okay? Because it's necessary to have all of those amino acids there to have that proper folding, okay? So there you go. There's one example there. Okay, again, they have other sort of jobs as well. So also stabilize unfolded polypeptide chains during transport into organelles, okay? So we've been talking about, proteins are made on the ribosome, but then they have to be transported into that appropriate part of the cell where they're going to be used, right? So for example, it says partially unfolded proteins are stabilized by chaperones, basically as they are transported across the mitochondrial membrane. So obviously these are proteins that are going to be used in the mitochondria, okay? They're made in ribosomes in the cytoplasm. However, they need to get into the mitochondria, okay? And then it says chaperones in the mitochondria then facilitate folding, okay? So if you look at a picture, this is what this is showing you. So again, here's your polypeptide chain associated with these chaperones in the cytoplasm, right? However, it's not going to function in the cytoplasm. It needs to get inside the mitochondria. If it is folded up like this on the outside, there's no way it will be able to be transported in, right? So these chaperones keep it in an unfolded conformation and target it to the mitochondria where it gets transported in. And then you'll see once it enters into the mitochondrial matrix here, there's other chaperones that then help it properly fold inside, okay? So not until it's in this state is it functioning. So you guys see that? So these are chaperones out here, but they're different chaperones than the ones that exist in here, right? Okay. A little more about chaperones here. So these guys were initially identified as something called heat shock proteins, and they're abbreviated HSP. And so basically they saw that these heat shock proteins were expressed in cells that were subject to high temperatures, okay? And then maybe at the time when they first discovered them, they didn't necessarily know exactly what they did, right? So heat shock proteins will stabilize and facilitate the refolding of proteins that have been partially denatured, right? So if you heat or cells are subject to high temperatures, their proteins are going to start to denature, right? And so that would then activate sort of this process or the expression of these heat shock proteins to help those proteins, okay? So specifically, there's a heat shock protein 70 chaperones, and also chaperonins, we'll talk about them in a second, are found in both prokaryotes and eukaryotes, okay? And so the job of those types of heat shock proteins is to stabilize the polypeptide chains during translation and basically transport them, okay, where they need to go. But bottom line is that sort of the heat shock protein name, or they got that name because they saw high expression of these types of proteins when cells were subject to high temperature. Okay, so I just mentioned this on the last slide, but we've been talking about chaperones. Well, there's also chaperonins, okay? So again, if you look at your review sheet, you'll see the first thing that's listed there, chaperones. Heat shock proteins are listed there because those are a type of chaperones. We talked about kind of just basically what chaperones are doing during translation, right? That they're binding to that polypeptide chain as it's made to make sure that it does not fold improperly until the full thing is made. Okay, the next thing on the list here is chaperonins. Okay, so the polypeptide can be actually transferred to something called a chaperonin. If we're talking about so the one example I showed you in the mitochondria, right? This guy's unfolded here and it folds properly inside the mitochondria. So think about with the chaperonins as them being proteins that maybe are going to function in the cytoplasm, okay? So it's kind of like this sort of barrel-shaped structure, I guess, where the actual the polypeptide chain will enter inside, and that will isolate the protein from the cytosol and from other unfolded proteins and allow it to fold properly, okay? So here again, you see this heat shock protein 70, which we were talking about, right? That we see both in prokaryotes and eukaryotes. And what it does is, again, it associates with that growing polypeptide chain, okay? And basically, it sort of brings or allows for the transfer of this not yet folded polypeptide chain to go inside, I said, kind of this barrel-shaped protein here, the chaperone, and allow it to fold properly. And the reason why is that it sort of provides a separate compartment for that protein folding to happen appropriately. And so it provides an environment that's slightly different than the cytosol. It also keeps it away from other unfolded protein. So if there's other misfolded or unfolded proteins around, the proteins can aggregate together with each other and it can cause problems. And we're actually going to talk about that in a second, okay? There are diseases that are, believed to be caused by defects in protein folding, right? So say there was a defect in the chaperonin, and this was not able to enter inside. Well, again, what might happen is that it might be misfolded, and then it may aggregate with other proteins around. Therefore, they're not going to function. They pool up inside the cell, and they can cause cells to die, okay? So, and we'll kind of talk about this a little bit more along the way, and I'll actually show you on Wednesday, I believe, or I actually may pull it. Maybe I'll pull it up here. It depends on how long this takes. But at some point, or if you look on Moodle, there is a CTE review paper, okay, there that kind of it's related to misfolding of proteins, okay? So anyway, we'll kind of follow up and have a little more on this as well. And I believe there may be another slide or two that talks about this a little bit more as well. Okay, so this is mentioning here cystic fibrosis. So it's caused by a mutation, right, that results in one amino acid deletion that leads to improper folding of a chloride transporter, right? So a chloride channel. And we've talked about cystic fibrosis a couple times. And so basically it's a defective chloride channel, okay? But the defective part, because of that one amino acid that's missing, it leads to defective folding, okay? And so if that chloride channel is not folded properly, it can't function, okay? Alzheimer's disease. So yeah, so I knew I was going to mention this. I didn't know how soon it was. I could have just flipped the slide and figured that out. So here, again, if you look at your review sheet, you'll see it says diseases caused by defects in protein folding, right? So we gave an example of cystic fibrosis. Here's another example. Alzheimer's disease, Parkinson's disease, type 2 diabetes. I told you CTE, which I'll mention a little bit later, or mention that another time in class or whatever, or during our Zoom meeting. They are all associated with aggregation of misfolded proteins, okay? And so what happens to those misfolded proteins, they'll form aggregates called amyloids, and so they're sort of characterized by these beta sheet structures, okay? And so here you can kind of see this amyloid state of these proteins because they're misfolded and then they aggregate together like I talked to you about, okay? And actually, if you look at, you can see these, what you call them A-beta, amyloid beta plaques in Alzheimer's disease, right? So this is showing you brain tissue, and you can actually see these aggregates that form these plaques within that brain tissue. And you see similar plaques like this in CTE, which is chronic traumatic encephalopathy, right? Which is caused by repetitive hits to the head or repetitive concussions. Okay, so again, we'll follow up a little bit more on that. Okay, but again, all caused by misfolded proteins. Okay, so again, here are some other examples as well. So CTE is not listed on here, but we'll talk a little bit more about that. So what this is showing you is actually the type of aggregating protein. So Parkinson's, so Alzheimer's, I told you A-beta plaques. That stands for the amyloid beta because that's the type of protein that aggregates. Parkinson's, it's alpha-synuclein, if I could say it, right? Huntington's, the protein is Huntington. So there are a lot of diseases that are caused by misfolding of proteins, and then as a result of that, they aggregate together, and you end up having cell death. Okay. All right. Oh, there's a little bit more on this. I think I kind of already said this. So amyloid plaques, right? I told you those A-beta plaques are aggregates of that A-beta protein. Also, you'll see these tangles, which are caused by misfolded tau proteins. And that tau protein is the protein that we talk about when we talk about CTE, okay? So this is showing you the plaques, and this is showing you the tangles. And so again, this is brain tissue where you're particularly staining for those particular proteins, and you can actually visualize them on a section of brain tissue. Okay. All right. Also, something that's associated with misfolding of proteins, something called prions, right? And so these are not living things themselves, but they're misfolded proteins that actually can self-replicate. And they also then will induce other proteins to misfold as well. Okay, so some diseases that are caused by prions, mad cow disease, right? There's another called scrapie in sheep, Creutzfeldt-Jakob disease, Kuru. Those are all diseases that are caused by prions. Okay, again, all involved in the folding or misfolding of proteins. Okay, so this is showing you actually how these prions can sort of aggregate and sort of lead to, they can basically self-replicate. So basically you're obviously augmenting the problem, right? And creating more and more of these misfolded protein aggregates. Okay, so we sort of move on from there. So we talked I give you quite a few examples about diseases caused by defects in protein folding. And I would expect to have some sort of essay on that on the exam, right? And maybe one that you kind of need to think about a little bit more. And I may ask you to really describe to me how protein folding is involved in one of these diseases, right? And explain, how it leads to the pathology that you see. Okay. In terms of, some details, okay, about the folding and stuff here. Okay, so these are just some enzymes that are involved, right? So it says 2 enzymes can act as chaperones by catalyzing protein folding, right? So these are in addition to the chaperones we've learned about before, okay? Those other chaperones that we talked about before weren't enzymes, okay? So that's the reason why we're just mentioning these guys here. But these are other types of chaperones. So protein disulfide isomerase, or PDI, will catalyze disulfide bond formation. So, when we talk about the tertiary structure of the protein, we talk about the presence of disulfide bonds, okay? This enzyme itself, which is technically a chaperone because it helps with the folding of proteins, is involved in catalyzing those disulfide bonds. Okay. All right. So it says it's abundant in the ER, the endoplasmic reticulum, where basically there's an oxidizing environment, and that basically allows for those disulfide linkages to occur. Okay. So a lot of protein processing and sort of modifications and folding actually happens inside the endoplasmic reticulum, the ER. And we'll talk more about that in a following chapter, okay? All right, so this is just showing you here, right, that this is an enzyme. This is a chaperone, but it's actually an enzyme that catalyzes the formation of this disulfide bond, which is important in forming that final conformation of that protein. Okay, here's another example of one. So peptidyl-prolyl isomerase will catalyze the isomerization of peptide bonds that involve proline residues. And so this is actually also, again, another enzyme involved in forming different types of linkages that are involved in protein folding. Okay, I don't expect you to memorize each of these specific different enzymes. They're just meant to give you examples of other types of chaperones. Okay. All right. Okay, so this is just showing you that there. Okay, so some more processing, okay? So all of what we just were talking about was protein folding, right? We talked about chaperones, chaperonins. We talked about diseases that are associated with defects in protein folding, okay? And then I gave you just 2 examples of other types of chaperones that act as enzymes, okay? But bottom line is, know what chaperones are. They are proteins that help other proteins fold, and they can do it in various ways, okay? In terms of more processing here, this is protein cleavage, right? So proteolysis, right, is the cleavage of a polypeptide chain, okay, which will remove portions of it, like that initiator methionine, okay? So sometimes these proteins or these polypeptide chains need to be cleaved in a certain way before they can be functioning proteins, right? Okay, so it says many proteins have an amino terminal signal sequence that will target them to wherever they need to go. That will need to be cleaved off before it can function the way it needs to. Okay. All right, so I'm gonna show you a picture of that. So this is showing you here. Again, this is about processing of proteins and particularly proteolysis, meaning something has to be cleaved off of that protein before it can function the way it needs to. Okay. So one thing that might need to be cleaved off, first example right here at the top, was that initiator methionine, right? Probably needs to be cleaved off. It's not that all that did was initiate translation, okay? Another thing that might need to be cleaved off from a protein is a signal sequence, okay? So this signal sequence in red, its only function is basically to target this protein to here, specifically the endoplasmic reticulum, okay? So it tells the cell where this protein needs to go. Now, as that protein or that polypeptide chain is being synthesized, it's basically entering into this channel here to enter into the ER. But as it's doing that, there is a enzyme that sits here that will cleave it off, okay? So again, what we're talking about is protein processing. And proteins need to be folded, right, properly. Sometimes proteins need to cleave things off before they are fully functioning, okay? If this signal sequence was left on here, it would not be able to fold properly and would not be able to function properly, okay? All right, so more on protein cleavage, okay? So it says proteolytic processing includes formation of active enzymes, right? Or hormones by cleavage of a larger precursor. So there are many proteins that are basically made from larger precursors, okay? So I'll show you an example. So one example here is insulin, okay? It's synthesized as a precursor polypeptide that basically goes through cleavages, right? Basically gets cut up to produce that mature insulin, okay? So you can see here, this is what's made first, this pre-pro insulin. This isn't an mRNA sequence, this is the polypeptide sequence, right? But then there is a cleavage of a signal sequence, okay, that's cleaved off, right? So one thing is cleaved off, and you can kind of see this sort of initial forming, and then this gets cleaved off, from this proinsulin to get the fully functioning insulin. So there's kind of two rounds of cleavages that occur, or cutting out of certain areas of that pre-pro insulin before it can actually be insulin. Okay, so pre-pro insulin gets cleaved in a certain way, that signal sequence comes off to form pro-insulin, and then this whole red area gets cleaved off to form insulin. This is not functioning. This is a functioning molecule, okay? All right, another example, replication of HIV, okay? There is a virus-encoded protease which will cleave precursor polypeptides to basically form the viral structural proteins, okay? So if that cleavage of these precursor polypeptides doesn't occur, then those viral structural proteins won't be able to be made, okay? So this HIV protease is actually an important target in drug development, okay? So again, you can target this protease, which is specific to the virus, it's virus encoded, and it will stop those polypeptides to basically be, the larger polypeptides to be cleaved to basically make those fully functioning structural viral proteins, okay? All right, now more on processing, okay? So those were all examples of cleavages, right? So, or ways in which, ways in which those proteins are basically cut up, or parts of them are cut out before it can be fully functioning, okay? So hopefully you're still following along on your review sheet, okay? So again, if you're looking under, so we talked about proteolysis, right? Signal sequences, it says cleavage of larger precursors like insulin. And now another way in which proteins are processed is glycosylation, okay? And we've heard the term, or remember the term glycoproteins. And so a glycoprotein is just a protein with a carbohydrate group attached, okay? And so the protein's made and then it has to be processed, meaning the carbohydrate has to be added on. Okay, so that's what we're talking about, processing. Things that are added on to the protein, right? Or things that are cut out. All these modifications that happen to that protein or happen to that polypeptide chain after it's made. Okay, all right. It says the carbohydrates that are added to proteins, right, to form glycoproteins are important in protein folding. In the ER, I told you a lot of protein folding and processing actually occurs inside the endoplasmic reticulum. It's also important in targeting proteins for transport and also as recognition sites of cell-to-cell interactions. Remember, they're glycoproteins that sit in the plasma membrane, right? And that those carbohydrate chains stick to the outside of the cell. Okay? Now, there are different types of glycoproteins. To be honest, you guys don't really need to know the specific details here, but I want you to be aware that there are different types of glycoproteins, okay? So there are N-linked glycoproteins because the carbohydrate chain is attached to the nitrogen atom on the side chain of specifically asparagine. O-linked because the carbohydrate is attached to the oxygen on the side chain of serine or threonine. Okay, so we should know what the difference is between the two, and the name kind of helped you with that, right? So this is just showing you there. So again, I don't usually and probably will not, ask you to identify these things from a picture. Why are you eating that? Sorry, my dog's eating random stuff. I don't know where you got that from. Okay, but I want you to know what those are. So if I said N-linked, we should know what that is versus an O-linked glycoprotein. Okay. All right. In terms of glycosylation and kind of how it happens, right? To just give you a little more detail into that. Again, it says it starts in the ER before translation is complete. Okay. So let's see. Let's look at a picture here. Nobody, again, needs to know the exact mechanism for how these things are happening, okay? But let's kind of see a picture to see how this process occurs. So here's your mRNA. That ribosome is basically targeted on the ER. Remember we talked about the ER, the rough ER, right? It has ribosomes sitting on them. Well, those are going to make proteins that need to go inside the ER. That's why they're sitting there, okay? So here's your polypeptide chain being made. It's going right into the ER. And as it enters into the ER, right, you have this carbohydrate chain sitting there ready to be added, okay? And basically, it gets added, okay? Obviously, there's lots of things that are happening, in between here and here that we're not talking about, but it's going to be added inside the ER. And it's sort of simultaneously as it's finishing being translated. Now, those were N-linked, right? Glycoproteins, ER. So O-linked carbohydrates, right, are added within the Golgi. Okay, so this is showing you here. Right, so this is showing you O-linked examples, and that's pretty much where we'll leave it. So I'm not even going to show you the specifics there. Okay, so we gave an example of N-linked and O-linked in terms of them being glycoproteins, right? Where the carbohydrates are added. So take home, we need to know what a glycoprotein is, okay? Understand that if we talk about an N-linked versus an O-linked, it just is describing where the carbohydrate chain is attached within the protein, right? And if we talk about when or where or how those carbohydrate groups get added on, right? Some of it will happen in the ER, and you can kind of see how the carbohydrates basically sitting there, sort of waiting, right? So these are N-linked. As it's being translated, it will essentially it basically moves into that ER as it's being translated, and then that carbohydrate chain will be added. And so glycosylation is the addition of carbohydrate chains, right? Write it on your review sheet, to proteins. So we're talking about how are proteins modified? Well, some proteins need to have carbohydrate chains added onto them. That's called glycosylation. We call those proteins glycoproteins. That's where these names are coming from, okay? This was giving an example of how or where it's sort of added on, okay? O-linked ones are added within the Golgi instead of in the ER, okay? Don't worry about how they're formed, okay? This is just showing you an example. Don't worry about it. Okay. Another thing that needs to happen sometimes to proteins is that they need lipids added. Okay? So some get modified by the addition of lipids. And again, you can see that right after glycosylation in your review sheet. Now, there are different types of lipid additions, okay? And these are examples here, okay? Again, don't memorize these, different types here. I just want you to be aware that there are different types of lipid additions and that they're specific kind of to where they function, right? So these guys are associated with the interface of the plasma membrane, okay? And this is actually showing you how we're adding this on, okay? So myristoylation, right? So myristic acid is what's added on, which is a 14-carbon fatty acid, okay? You don't need to know these details. So I'm basically, you don't even see it on your review sheet. So hopefully you listen to this and you know that you don't need to worry about the different types of lipid additions, right? So again, you can see another type, prenylation. So prenyl groups are added. And again, the reason why we're talking about these different groups is because you can see they have specific sort of functions, right? So these type of proteins with prenyl groups on them are involved in the control of cell growth and differentiation, okay? Some can actually be responsible for human cancers. And so this is giving you an example or showing you that. Another type, right, palmitic acid, which is a 16-carbon fatty acid, can be added. And this has to do with the association of those proteins with the cytosolic face of the plasma membrane. Okay, another one, glycolipids. Okay, so again, these are just some examples. Just know that lipid additions, and maybe make a general statement kind of on your review sheet in terms of why lipid additions are important, okay? And so you can pull just some of those functions that I just mentioned. You don't necessarily need to know that prenylation is the one that's mostly associated with those proteins involved in cell growth, okay? But I just want you in general to understand the importance of lipid additions per se, okay? All right. Okay, so we can move on from there. All right, so now we're moving on to regulation of protein function now. So that was all, again, what we all were talking about a lot of the last, there's quite a few slides, right, was protein processing, okay? We talked about protein processing or protein folding first, right, and the involvement of chaperones and chaperonins. And then we talked about the cleavage of proteins, right, proteolysis. And then the addition of things like carbohydrate groups, right, or lipids being added and how that's going to affect the function of the protein. So just to continue and to try to finish up this chapter, let's talk about regulation of protein function, all right? Obviously, we know that the proteins are the functional units or the functional molecules of the cell, okay? So the cell must have ways of regulating proteins. And I think we've already talked about some of these ways just throughout the semester, okay? So 3 main mechanisms: regulation by small molecules, phosphorylation, we already talked about, and other modifications, and also protein-protein interactions. Okay, so what we mean by small molecules, we've talked about this before, right? Like feedback inhibition and allosteric regulation. Okay, so if you think about enzymes, they're often controlled by a small molecule binding to an allosteric site, right? A site other than the active site, and that regulating its activity, okay? And so we talked about this before. Here's that same example we've seen before. Here's your precursor. Here's first enzyme, second enzyme, third enzyme, okay? That end product of this metabolic pathway can bind to an allosteric site and basically inhibit this, okay? So that's a small molecule that's regulating a protein. This protein happens to be an enzyme, okay? Also, it doesn't always have to be an enzyme, right? And that small molecule doesn't always have to be an end product of a metabolic pathway, okay? And we've mentioned this already. That many proteins, we saw this example with the translation with the initiation factors, right? That they're regulated by the binding of GCP or GDP, small molecules binding to it, okay? There are a particular class of proteins called the RAS oncogene proteins that are regulated that way, okay? So just the binding of either GDP or GTP will make that protein either be active or inactive. Okay, so we already saw that example as well. All right, so this is particularly giving you more information about those RAS proteins. Okay, so mutations in RAS genes is believed to contribute to about 25% of human cancers. Okay, and so those RAS proteins can basically be altered and locked in the active position, and basically they will keep continuing to signal cell division to happen and stimulate cell division, right? Stimulate proliferation of cells. Okay, other modifications, right? Phosphorylation. We talked about this already. Phosphorylation, totally reversible, right? It can activate or inhibit proteins depending on what's going on. Kinases are proteins that will add a phosphate group. Okay, so that's what this is saying. It transfers phosphate groups from ATP. So basically it takes a phosphate group from ATP and it adds it to a protein. Where it adds it, right, is basically hydroxyl groups to the side chains of serine, threonine, and tyrosine. So that's telling you particularly where those phosphate groups are usually added. Phosphorylation can be reversed, meaning there are proteins that will dephosphorylate or remove a phosphate, and those are called phosphatases. You should know what a protein kinase does and what a phosphatate does. Okay, so a kinase adds a phosphatase removes a phosphate. Okay, again, I think we talked about this already. All right, and so this is what this is showing you. It's saying it's a serine threonine kinase because that's where it adds the kinase to a serine or a threonine in that protein. Okay, here's a tyrosine kinase because it's going to add it to the tyrosine in that protein. Okay, all right. Okay, it says protein kinases are often components of signal transduction pathways. And we will spend a little bit time, we don't cover the entire signal transduction chapter or anything like that, but we do cover parts of it. Okay, and I think we gave this example already as well in a earlier chapter. Okay, so it says, what's a signal transduction pathway? Sequential action of a series of protein kinases, meaning something binds to the cell surface and it activates a whole bunch of other proteins inside that cell that essentially is a sort of cascade of events, okay? And it then will result in changes in that cell based on what's happening in the environment, okay? So the example, right, muscle cells, epinephrine will bind and it will signal the breakdown of glycogen to glucose, okay, by basically activating a specific enzyme, okay? That's what this is telling you here, right? We saw this before. Hopefully I gave you the same picture. So it's a little bit different, but maybe not. Maybe it's the same exact picture, but essentially same thing's happening here. Here's epinephrine binding to that muscle cell, right? Series of things happens inside the cell. And then basically you have a particular kinase that gets activated. That kinase then will phosphorylate a specific enzyme, which activates that and then leads to glycogen being converted into glucose. Okay, so we saw this example before. What we're now focusing on is really this phosphorylation, right? And that the cell uses phosphorylation as a way to regulate the activity of proteins, okay? Kinases, right, or a signal transduction pathway is this, what you see here, this series of events that happens, okay? Kinases will phosphorylate, phosphatases will dephosphorylate or remove a phosphate, okay? All right. It says aberrations in signaling pathways, right, especially in protein tyrosine kinases, are responsible for some cancers and for other diseases as well. And so the first protein tyrosine kinase that was discovered was actually in 1980 and was from this particular sarcoma, okay? And so there is a key experiment, and I probably have it up on Moodle, and so if we have time, I will draw your attention to it, but to save time, I'm not going to pull it up right now. But basically, there are small molecule inhibitors of these particular enzymes that actually have been used to treat cancer, okay? All right, other types of modifications that can regulate protein activity, acetylation, methylation, okay, glycosylation. So there are other sort of additions that can regulate activity as well. Okay. Okay, so now we're going to talk about Well, I guess, yeah, I guess this kind of ties into, now we're going to get to protein degradation. Okay. So one little extra thing here. So it says some proteins are regulated by covalent attachment of polypeptides. So other proteins that will interact with them. And so that's kind of the third thing on the list there under regulation of protein function, protein-protein interactions. Okay, so it says addition of ubiquitin will target proteins for degradation. Other types of proteins like SUMO can affect other functions as well. So we're going to talk about protein degradation in a second. But bottom line is proteins can be regulated by other small proteins binding to them. Okay. All right, so that's what this is saying here. Histone modification by ubiquitin and SUMO is one mechanism for regulating transcriptional activity of chromatin. Okay, ubiquitination, ubiquitination, right, or adding ubiquitin is also important for regulation of protein kinases. Okay, so addition of, and there's sort of certain proteins that are more likely to be used as sort of regulatory proteins, right, like ubiquitin and SUMO, and when they're bound to other proteins, they will alter their activity. Okay, and so these are some processes that they're involved in. What we're going to talk about next is actually protein degradation. And actually ubiquitin is involved in this as well. Okay, so I guess the idea is not every ubiquitin is not only always associated with the protein degradation, but it can have other effects as well. Okay, so it says protein levels in cells are determined by rates of synthesis and rates of degradation, right? So we can talk about half-life of a protein. And what do we mean by a half-life? The time it takes for half of that protein to be broken down, okay? And so obviously, I think I mentioned this kind of at the beginning, obviously if a protein has a really long half-life, it's going to stay around longer and therefore function for a longer amount of time and be more active. Something has a really shorter half-life, we would say it's less active, okay? So it says many regulatory proteins have shorter half-lives because they need to be able to be degraded quickly and then allow that cell to respond to what's now happening in the environment, okay? Faulty or damaged proteins will actually be recognized and degraded on purpose, okay? So like I said, the major sort of pathway to do this is something called the ubiquitin proteasome pathway, and you'll see that under protein degradation in your review sheet, okay? So ubiquitin will attach to a protein. So now think about a protein that's maybe misfolded or misfunctioning. Ubiquitin, or the cell will identify it, and ubiquitin will sort of be that identifier. And so once ubiquitin is added, usually other ubiquitins come along, and it says it's polyubiquitinated, meaning it has multiple ubiquitins bound to it. They get recognized and they're degraded by a large protease called the proteasome. So think of the proteasome as a whole bunch of proteases or a whole bunch of enzymes that will chew up proteins, and that function is basically to break that protein down and get rid of it because it's not functioning properly. Okay, that's what this is saying. So polyubiquitin, you can't say that, right? Polyubiquitination, right? So meaning has multiple ubiquitins added onto it, signal that protein to be degraded. Remember I told you before that ubiquitin sometimes is used as a regulatory protein, not necessarily always as part of this pathway. So think about if you have multiple ubiquitins on it, it's going to target that protein to something called the proteasome, which is a whole bunch of enzymes in there that are going to chew it up and destroy it, okay? All right, so it says many proteins that control fundamental cellular processes are targets for regulated ubiquitination and also proteolysis. So cyclins that regulate progression through the division through cell cycle, right? That basically tell that cell to move from, G1 to S to G2 and move through. Cyclins are those proteins that regulate that. And they regularly get degraded, and then so they sort of accumulate and then they get degraded, and that's what signals that cell to move on. Okay, so the point is that is a sort of normal process that's going to occur. Okay, and so if you want to talk a little bit more about this, that's the last I guess it's not the last part. Maybe we'll talk about autophagy for a second too, but this is involved. Okay, so degradation and the cyclins, right? So let's look at a picture here, okay? So I guess you don't only want to just think about the proteasome and a protein being degraded as sort of, I don't know, associated with a negative thing, right? And that it's not functioning properly. That's not always the case. Yes, that's the case sometimes. But sort of normal functioning and normal progression through the cell cycle. This is actually showing you through the different phases of the mitotic phase even. There are certain proteins that need to be degraded in order for that cell to move through. Okay, so say here, if you look here, this is the cyclin B CDK1 complex here. If this cyclin B doesn't get degraded, well, one, it doesn't get targeted by the ubiquitin first and then degraded, this cell will never be able to move into anaphase. Okay, it will be stuck here. So the degradation of the cyclin B is really important to basically allow that cell to cycle through those phases that it needs to go through. Okay. Okay. Another thing to talk about. We talked about lysosomes before, right? Hopefully that term sounds familiar. And basically, you always say it's the sort of digestive system of the cell. And there are proteases in there. Okay, there's digestive enzymes, including proteases, that break down proteins inside the lysosome. And so those lysosomes can digest extracellular proteins that are taken in by endocytosis. And basically, also, though, they can engulf organelles and other intracellular proteins that need to be degraded as well. Okay. The reason why those digestive enzymes need to be confined to the lysosomes is that if they weren't, they would just degrade everything, right? And some proteins need to stay intact in order to be functioning in the cells. Okay, so it says proteins will move into lysosomes by a process kind of called autophagy. Okay, and so autophagy is the idea of the cell breaking down its own cellular components, i.e. proteins or old organelles, on purpose, okay? And then basically recycling and using what results from it. So this is basically showing you, here's an old mitochondria that needs to be broken down. It's not functioning anymore, it's too old, right? And what happens is it basically gets engulfed by a phagosome here, autophagosome it says, and then that autophagosome will fuse with the lysosome. Why? Because that's where all the enzymes are, those proteases are, to break this down, okay? And then it'll basically be broken down there. Okay, so autophagy, we'll come back to that at some point, but that's a normal process that occurs, and it's basically the cell breaking down its own constituents. Obviously, if this is out of control and too much occurs, it can lead to the cell dying, right? If it doesn't occur enough, you're going to end up with misfunctioning organelles and misfunctioning proteins. Okay, so it's a process that the cell has to get rid of old components that it doesn't need. And then once it breaks this down, it can use, say, these amino acids that are there to actually remake other proteins, right? And other organelles. Okay, so here is kind of that autophagy, a little more on that autophagy, which I was talking to you about. Okay. Right? Autophagy is activated during nutrient starvation. It allows the cell to degrade non-essential proteins and organelles so that it can reutilize those components. Okay, but again, it's a normal process. Yes, you see it, I guess, activated or increased during nutrient starvation, but it's always happening, and it's healthy. It's a normal process. Okay. Finally, we are done.